to open up our afternoon proceedings, we have Matthew Blumberg. So Matthew Blumberg works in the field of network computing and large-stage collaboration. He is also working on Dalya DAO, which is going to be a neglected disease DAO. And yeah, we're super excited to have him here. Sorry, a mic would be useful.
Hi, everybody. A few more people coming in the door here. So I'm going to talk to you today about a set of 3 interrelated projects, which collectively you could describe as a sort of tool stack for the work we aspire to do in developing impactful drugs and bringing healthy life to as many people as we can. So the first is Charity Engine, a key bit of infrastructure, which is a crowdsourced cloud service, we call it. The precursor to this was a project of mine called Grid Republic. And with Grid Republic, it's a sort of classic citizen science volunteer computing thing. We have a little application that we give to as many people as we can, and it allows them to donate their computer spare processing power to, in this case, scientific and medical research. And so we supported causes like astronomy, climate modeling, protein folding at one point, epidemiology, a wide range of things. This grew into Charity Engine, which is more properly now what we call the crowdsourced cloud, where we, in addition, run commercial work on the volunteered devices. So the value proposition for the volunteers is that we share the profits with charities that they care about, Oxfam, CARE, and so on. And we also donate a considerable portion of the compute resource to scientific and medical research. At this point, our grant program has donated more than 1.5 billion hours of compute time to scientific research, including, we're proud to say, over $200 million to the lab of David Baker, who recently won the Nobel Prize for his work in protein structure prediction. The network has about 1 million CPU cores, 100,000 GPUs, several petabytes of storage, so it's an extremely capable resource. In terms of geeking out, just very briefly, these resources are provisioned to users as standard instance types. You can just use any Docker container. There's simple interfaces like an API, a CLI, a web UI. You can run your compute jobs through a smart contract, although nobody actually ever has. And we have a nice integration with Wolfram Research, so you can run jobs from Mathematica and Wolfram Engine directly onto our network. Some projects that we've run in the past we had a really great finding with some researchers at MIT and Bristol University solving a long-standing math problem. It took about 35 million CPU hours, which we were able to provide them in a couple weeks. We did a huge genomic search project where we had genomics data spread over 350,000 devices so that we could search a gigantic data set really fast by parallelizing the disk I/O, basically. And we're running a project with CDC in the US, that's the Center for Disease Control, where we have sequence data from environmental samples around the world submitted daily, which we screen for a range of pathogens of concern to them as a way of early detection of biological threats. So it's an extremely powerful compute platform. If anybody doing BioDAOs has need for large-scale batch computing, please do get in touch. But mostly it's built, and actually from the beginning was built, to facilitate our work in the area of drug discovery. Which brings us to Find.bio. At Find.bio, we describe what we do broadly as reverse engineering biology, but the goal is to pursue a range of strategies for computationally augmented drug discovery. So in order to explain how that works, I want to just take a step back and explain what drug discovery, what biology looks like from the point of view of structural biology. So the basic idea is that a cell is sort of a fantastically complicated but fantastically tiny machine in which all the parts are substantially made out of proteins. So you have little protein gears and little protein motors and things like this. And this is actually not a metaphor. Like literally, there are little protein gears, and this is a rendering. This next one is DNA replication at the molecular scale, right? There are these little mechanisms at work in your body, and they're super tiny, and there's just huge, colossal numbers of them. And the basic idea is that a disease is a malfunction of the machine. So those proteins, the parts of the cell, the parts of the machine, their function is determined by their shape. So if one gets misshapen or it's missing, then it can cause a cascade of events that are manifest at the large scale as a disease or a pathology. So when you want to fix a broken machine, the first thing you have to do is, well, you figure out what's broken, like what part is broken. And then you can try and fix it. And typically drugs, this is what they do. Drugs are basically either they try and fix a pathological change, or equally, maybe more often, they just try and break it completely. And that interrupts the pathology, kills that cell, and a healthy cell tends to replace it. But again, if before you can try and fix it or break it, you need to know what part is at issue. So virtual drug screening is a method of drug discovery. It starts, in this case, when you've already determined a drug target. So for instance, there's a wide range for diseases you might be interested in. Early stage research is all about figuring out what's broken. And they'll typically name a protein that's fundamental to the pathology, and they'll call that a drug target, by which they mean this is something you should, when you're developing a drug, the thing you're trying to target. So you might have, when you're trying to undertake such a discovery program, different amount of knowledge that you're starting with. You might already know some compounds that interact with your target, in which case this is really great because now you're in an area where you're just trying to optimize them in various ways. You might know enough about the target to know where on the target you want to be interacting, or you might only know that this is the target you want to address. So in what I'm going to talk about moving forward here, we're going to focus on the second case, because if you're in the first case and you already have some known compounds, you're well ahead of the road. There's still some fascinating things you can do to optimize it computationally. And if you're in the third case and you know just the target, there's some preliminary work that you would do that's reasonably straightforward that would get you to number 2. So this is sort of like the base case for virtual screening. So virtual screening uses a method called molecular docking. And what that means is the process of taking very large libraries of compounds, one at a time, and testing them against your target to see which one will bind effectively. So by way of illustration, we have here illustration of a protein target. And you might search through, sometimes it's described as like a lock and a key. The protein is a lock and the small molecule is a key. So you're searching through, trying to find a molecule that will fit. Look at, there's one. And that would be potentially a drug candidate. So here's a 3D model of a protein, and this red marks the binding site. And again, you look through to find molecules that you think, or that will bind to it. And so here's an example of one. It really gets in there. That would be a really strong drug candidate because it gets in there, it fills up the volume, it's going to get stuck. So that's what you're looking for. And here you see a search. All right, there's lots of different molecules, and it's exploring the surface of that protein. And so docking is you would search very, very large numbers of molecules. But the search space for such things is extremely large. The size of chemical space is about 10 to the 60th. So for context, 10 to the 24 is the number of stars in the observable universe, and 10 to the 17 times 4 is the age of the universe in seconds, right? That is really big chemical space. And it's worse because the chemicals are very floppy, as are the proteins. So it's not surprising that what we're doing with docking is a brute force approach where we're just looking through huge numbers, that the scale of your search really does matter. And so you see here that a comparison of screening 99 million versus 1.7 billion. 1.7 billion is sort of about the state of the art at the moment. And you see that a number of features of the process are, the results are much better in terms of how many hits you get, how effective they are, and the novelty of them, which is important. You want many different kinds of compounds in your set. So here, going back to the earlier conversation about Charity Engine, our compute network gives us the ability to screen around 10 billion compounds, which is 5 times more than the state of the art, which can be expected because of the higher resolution of the screen of chemical space to get us really best-in-class results for our drug screens. I'm going to skip this part where I talk about how you can even discover the targets themselves from early stage research, because I think time might be shorter than I thought. But oops, just to point out in context of the earlier conversations, what we have built is essentially an agentic workflow for doing this kind of work, where we have a number of modeling agents, some that model small molecules, some that model proteins, a simulation agent, and then machine learning agents that aggregate molecular information and simulation results to make useful predictions. So now I want to speak briefly about Dalya, which is a DAO we're launching to do neglected disease drug discovery. Neglected diseases here is not rare diseases. Neglected diseases are diseases that impact mostly people in poor countries and consequently don't attract R&D funding. So this chart shows just broadly for context the burden of disease worldwide and splits it among communicable infectious diseases versus non-communicable diseases. And the metric here is what's called a disability-adjusted life year. Years of healthy life that are lost due to disease. And so one DALY is one lost year of healthy life or productive life. What we see here is that if you break down a little bit, you see that the distribution is different around the world. So on the left, you see in wealthy countries like the US, very little infectious disease and mostly non-communicable diseases. But if you look at on the right, which shows what happens in poor countries, there's an enormous burden of infectious disease. And if you drill down a bit further, here you see particular categories of diseases. You see that on the right, basically 1 in 5 DALYs lost worldwide every year are in these 4 causes on the right, and they barely appear on the left. If we reformat that in a different way, some of the earlier chart, on the left, you see the poorest billion, again, getting a high proportion, the others were absolute numbers, of infectious disease, and on the right, high-income countries, it's mostly non-communicable. And now if you look over here, and you look at the proportion of drugs that have been developed in any phase of clinical development from 1995, you see it looks almost exactly like the proportion of the disease for the rich countries, which is to say that the pharma industry is serving their wealthy clients and nobody else. So it's to the extreme that only 1% of the drugs developed since 1995 have been for neglected diseases, which affect 2 billion people a year, and they have had almost no drugs since 1995. It's like embarrassing. It's I mean, the scale of the crisis compared to the amount of the research establishment's attention to this is completely out of whack. So the question is why? You know, like, well, there's a lot of people. Why isn't anybody making drugs for them? And the answer is because the pharma business is a business, and management has an obligation to maximize return to their shareholders, and so they would get fired if they developed drugs for poor people Not because they're bad people, but that's because how our economy works. They have to make money for their shareholders. So we're introducing Dalya, and we want to try and leverage the power of crypto to create a different kind of economy. So we say at Dalya, our currency is time, and by that we mean that we're going to value our portfolio of drugs not by the revenues they produce, but by the years of healthy life which they save. And so when you buy a Dalya token, you're saying a year of healthy life has value, and I believe that, and I'm going to put down some money on that conviction, and we will try and maximize the years of healthy life that we can deliver. So broadly, a regular pharma company will value its portfolio by taking the addressable market of each of its drugs, and at various stages saying, what's the probability of success of this drug? And they'll claim a percentage of the addressable market based on what they think their success probability is and how much time they have. We're going to do the same thing, but we're going to express our addressable market in years of healthy life. And as we de-risk our assets moving forward, the years of healthy life we can claim on our balance sheet should go up. And as long as our community continues to believe that a year of healthy life has value, then the value of our treasury should increase and give us the means to continue developing our drugs. So that's Dalya in a whirlwind. Thank you all very much.